Hi, everyone, and welcome back to the SIP here at Cal Academy. My name's Amy Jo, and today we're going to be talking to Gwen, who's one of the scientists here at the Summer Systematics Institute. So, Gwen, would you mind introducing yourself to us today with your name as well as your school and your major?
Hello, my name is Gwenndolyn Campbell. I go by she, her pronouns. I am currently attending Los Medanos College in Pittsburgh, California, over in the East Bay, and I major in biology and hope to transfer for botany.
Wow, that's very interesting, and it seems fitting. So, um, guessing that that's what you're studying this summer. So, would you tell us a little bit about what you're studying this summer?
I am studying Castanea, which is a genus of American wildflowers. They're native to the Americas and they are hemiparasitic uh, on other plants. And basically, the original idea was that we were going to try and differentiate better the species within them because there's a lot of variation and it's kind of hard to tell for a lot of scientists what exactly a particular specimen is species wise. And then it just morphed into something completely different that I'm really excited to talk about.
Great. So you used a term that I'm not sure everyone's familiar with. So would you mind telling us what hemiparasitic means?
Basically, it's a variety of parasitism within plants. A hemiparasite is essentially an organism that can um, essentially rely on a host for certain parts of its, water or nutrients or food, but can actually independently uh, go through its own cycle. It's not like dependent on reproduction onto these hosts.
Okay, great. And are these the flowers here?
Yeah, those are Castanea. These are several varieties within it. We have there's a lot of color differentiation within the genus, and another cool thing about the parasitism of these plants is that they actually basically connect to their host plants through the roots.
Wow. And so how are they parasitic through their roots?
Essentially, you can see through this Castanea tenuis diagram from the University of California that they essentially have these root structures underground, and they'll basically keep growing their roots until they hit a reasonable host plant's roots, and then they'll have these smaller tendrils that will essentially go into the roots of the host plant and draw out water or nutrients. It basically helps them better survive.
Wow. And are there like specific hosts that these wildflowers prefer?
That is actually a really awesome thing is that even within a single species of Castanea, they can be parasitic onto multiple different host species, like Castanea miniata, which is native to the San Francisco Bay Area, and they can be parasitic on species like coyote brush or yarrow or various other kinds. And you'll see a lot of variation within what host species are supporting these Castanea throughout their ranges, which, are basically from the Midwestern states all the way into the very west coast and also south. And they even have some varieties are in Alaska. So obviously the host plants will maybe be in one area but not in another. So that's sort of part of the variety of them.
Thanks. That's super interesting. And I know you mentioned that they're hemiparasitic. Are there any other
Mhm.
unique characteristics about Castanea that we should know about?
Yeah, actually, so these structures that you see right around here, kind of on the outline part, these are specialized leaves that will sometimes look like uh, another part of the flower. You can see it also over here. They're called bracts. And these are the most colorful and showy part of the plant. We call this the inflorescence, basically the width of all the flowers on the tip of the plant. But the colorful showiness is actually due to the leaves rather than the flowers themselves, which I think is a really awesome characteristic of Castanea. And also, there's just so much variety within the genus. You have varieties like this that have very rounded bracts and, have this color, but then you have ones that are sort of a lot more stunted and don't completely cover the flower itself and are a completely different color, or ones that kind of branch more outwards. I feel like one of the really awesome markers of Castanea is its variety, both in structure and color.
Yeah, it's easy to see that they're very varied amongst the varieties. Very, very cool. Thank you for sharing those photos. Um,
Mhm.
I wanted to go back and ask you, you said that sort of over the summer, uh, the, I guess the techniques for what you're studying Castanea this summer has evolved. How has that happened? Or what does that look like?
So, my mentor, Dr. Sarah Jacobs, she has been working with Castanea since, I believe, the time when she was getting her doctorate. Uh, so obviously she has this really wide variety in Castanea, and she specializes in speciation of different plants. And originally she was thinking we'd just be adding maybe to larger data sets, uh, that maybe we'd either be doing field work and getting more samples, or we'd just be going into the lab and maybe just doing molecular stuff where we're trying to sequence the genome of it. But then we kind of got into this really interesting thing called heteroplasmy, which is potentially present in some varieties of Castanea. It's basically a mutation where you'll find mitochondrial or similar DNA like chloroplasts from multiple sources within the cells of one singular plant. Um, Specifically, she wanted to look at whether paternal inheritance of chloroplasts was a thing widely in Castanea.
Oh, that's very interesting. And have you been able to do any work with that yourself in the lab?
Yeah, I've actually been doing about, I think, 14 samples from genetically distinct populations of different species of Castanea, and we're trying to basically look at genes that might show whether there's heteroplasmy present.
Interesting. And the heteroplasmy again is the, just to clarify, it's the chloroplast, the or what was it?
Hetero-
DNA
het-
like
Heteroplasmy would basically, it's kind of a broad term. It basically means that there is DNA from multiple sources within one part of a cell. Like, it can refer to the chloroplast, or it can refer to mitochondrial DNA that is only supposed to be inherited through the mother. Um, and basically, heteroplasmy would be the result of a mutation. And one of the things that Dr. Jacobs was trying to see was that maybe because chloroplasts aren't needed as intensely in these parasitic plants, that might be the cause of a mutation within that could cause the inheritance from, say, a paternal plant rather than the maternal plant, as would be typical.
wow. That sounds like very interesting work and you sound very like you've learned so much this summer. So thank you for sharing all of that.
I
Um,
have so much to learn still, but it's been an awesome experience.
Great. Have you learned any like practical lab techniques or any lab skills that you want to share with us?
Yeah, so I have basically been going all the way from getting leaf samples from Castanea all the way to extracting the DNA from that and then sequencing it with the help of a lab technician, Lynn Bonimo, who works in the comparative genomics lab here at Cal Academy.
Wow.
You'll see pictured before you the result of one part of this whole process. This is after we've tried to graft primers onto DNA to try and see if we can proceed to the next part of the process. We'll essentially fill a gel with this liquid that holds the results of one other part of it. And if you see that these small bars are lit up like this, that means that we have a good sample going and that we can proceed on to the next part of it.
Interesting. And what's written at the bottom?
This is basically the names of several of the samples I have. M3GC, which is the third sample that I've done, and M stands for molecular, the 3 is the number of sample, and then GC are my initials.
Oh.
Yeah, we went all the way up to, I think in this, the 10th sample, and then actually just this morning I was also in the gel doing the same step with my 11th, 12th, and 13th, and 14th samples.
Super. And
Yeah.
So what exactly are you looking for in these gels? You're looking for them all to light up. I know that.
Yeah. We're essentially trying to make sure that there is enough of our products, our PCR products, present in the gels that they light up really strongly, to know that, there's actually enough of that product present in order to go on to the next step, so that it's not just that we're loading up and trying to work with something that's completely blank.
Oh, okay. Interesting.
And then you'll see at both sides of these are what they call ladders, and these basically have like specific links of the pieces of the products in it so that we can kind of compare it with.
Very cool. Awesome. I wanted to segue and ask you, do you have any favorite places in the Bay Area to botanize in?
Oh, I've gotten so many opportunities even during this this internship to learn new places to botanize. This was at a place where you actually were here, Amy Jo.
Is this Pepperwood?
Yeah, this is the Pepperwood Preserve. So this is basically a nature preserve a couple hours north of San Francisco, where they have this group of people that are working to preserve this really unique landscape and trying to also create pathways for animals and plants to be able to connect more disparate ecosystems that they live in. So this is what's called a corridor area.
Interesting. And did you
And this
go ahead?
And I got the opportunity to botanize a bit over here. I was able to look at a lot of the endemic plants of the area and to kind of see I got to hear from so many amazing scientists over here, and that's probably why it's one of my favorite areas to botanize in, but not my absolute favorite.
Gotcha. And would you mind explaining what an endemic species is for everyone?
An endemic species is essentially a species that is unique to well, not necessarily unique, has evolved within a particular ecosystem or area.
Cool.
So, yeah.
What were some of the endemic species or endemic flowers that you might have seen at Pepperwood? Do you remember what did they look like? What were their characteristics?
Part of the territory in Pepperwood was chaparral, which basically is kind of similar to a almost deserty or prairie-ish brush sort of ecosystem. And then there are also these live oak forests. So those are some of the plants that would be present in those areas.
Super cool.
And then this was an area actually in Marin County where we had gone to find a Castanea group with Dr. Jacobs and my lab mate Cecilia Alvarado. This was really amazing. Also, a little bit of a difficult hike because we ended up going there a little bit late. So it was already starting to get hot up there, and the San Francisco Bay has that really nice layer of fog on it throughout most of the summer, but at this point it was just very hot.
Gotcha. Jumping off of that, I wonder if you have any tips for people who are looking for a certain flower. I know that throughout the summer you've been looking for Castanea, especially with Cecilia and Dr. Jacobs. Do you have any tips on finding any specific flowers?
I'd say that if you want to look for a specific species, then a really good resource to use is actually the iNaturalist app, which is worked on and developed by the California Academy of Sciences. It's basically this app that has crowd-funded data on the locations and variety of species of plants and animals in a given area. Particularly if you're in California, there's a lot of people all over the state that are contributing to data within it, but it's worth looking on no matter where you are.
Totally. Citizen sciences
We
is
love
on
citizen science.
it's on the rise. Cool. And I also wanted to ask you, do you have a favorite memory, perhaps from childhood or even the summer when you realized your love for science?
I'd say that one of the things that went into making me want to do botany specifically was an experience I had at the University of California Botanical Garden at Berkeley. Uh, I went there with my papa Mark, when I was, I think, 12 years old, and I was just struck by the sheer variety of different species of plants that the climate and the area in California could support. I was struck by the biodiversity of our area, and I just was all like, why shouldn't I try and study this? Why shouldn't I try and protect this? And I feel like that could be considered my personal genesis of why I want to do botany specifically, and also why I love science and biology so much.
Oh, that's beautiful. Thanks for sharing, Gwen. Well, I think we can sign off for today, but thank you so much for joining me, and thank you to everyone for coming to the SIP and listening to Gwen and hearing what she has to say about Castanea today. And we'll see you next week. Bye. See you,
Thank
Gwen.
you.
Bye bye.
Bye bye.